Good afternoon, everyone. Thank you so much for being here, and thank you for having me. My name is Mary Clay Bailey, and I'm one of the molecular pathology fellows at Baylor College of Medicine. And our home base for our fellowship is at Texas Children's Hospital, so that's where the data from today's presentation came from. So I'm really excited to talk to you about some of the work we've been doing about rare oncogenic structural variants in FGFR genes in childhood brain tumors and how they could potentially be effective therapeutic targets. Now it's my turn to figure out the diagram. Okay, great. I have nothing to disclose. And the first thing I want to go over is just how FGFR genes work generally, what they do when they're behaving normally. So FGFR genes are receptor tyrosine kinases, and there are 4 of them, FGFR 1 through 4. And as receptor tyrosine kinases, let's see if you can see, yes, great. So they have an extracellular receptor domain where a ligand will bind, and in this case, in FGFR's case, it would be the FGF, or fibroblast growth factor. And when it binds, this causes a dimerization and autophosphorylation in the intracellular compartment, and that activates the MAPK pathway, which is involved in cell proliferation and cell growth. And we will talk more about what happens when this pathway goes awry in a minute, but before that, I want to tell you how we selected our cases and identified relevant variations in FGFR. So the first thing we did was look at the Texas Children's Genomic Database between 2017 and 2024. And we identified CNS tumors specifically, and then even more specifically, the CNS tumors from this pediatric population that had oncogenic FGFR 1 through 4 alterations identified. So the way that these were identified and then reported out subsequently, we use an in-house solid tumor genomic panel. And for our in-house panel, we use NextGene and Platypus to call our single nucleotide variants, Archer for fusions, C and V kit for copy number variants, and then we use Pendel and Deli to identify the larger structural variants that are greater than 25 base pairs. After we identified the relevant cases, we did a deeper dive into the patient's charts and looked at clinical features and took another look at the histology and integrated diagnoses. Here's a more detailed view of the tumors that we selected. So out of all of the CNS tumors we identified in the time range, 29 of them had oncogenic FGFR alterations. So of these, this sliver here, this is broken down further. And the main thing that I want to point out here is that of the FGFR alterations, most of them were actually not single nucleotide variants. We actually identified more structural variants, over 70%. And this is a more detailed view of our data in a tabular form. So we'll start from the top and work our way down here. So the first thing to notice is that this table is separated into low grade on the left, low grade tumors, and then high grade tumors on the right. And these are individual tumor types that were identified and associated with the various FGFR alterations. So we have pilocytic astrocytomas, we had 6 of those, low-grade gliomas, there were 4, there were 6 dysembryoplastic neuroepithelial tumors, 1 rosette-forming glioneuronal tumor, 6 polymorphous low-grade neuroepithelial tumors of the young, and then 3 diffuse midline gliomas and 3 high-grade gliomas. And this row shows you the sex of the individual patients. So blue is male, and then the light green is female. Okay, so now we'll move on to the FGFR alterations themselves. The first one I want to talk about is FGFR1, and the red squares represent single nucleotide variants. So we only found single nucleotide variants in FGFR1. There were 8 of them. And then of the other alterations that involved FGFR1, we had 2 fusions and 9 duplications, and these were tyrosine kinase duplications. I'll talk more about those in a minute. And then for FGFR2, we mostly had fusions. There were 7, and then there was 1 amplification in a high-grade glioma. FGFR3 had 2 fusions, and then we didn't identify anything in FGFR4. So the bottom portion of this table represents the overall genomic landscape of each individual tumor, and the light pink squares mean that there's only 1 additional oncogenic alteration seen in these tumors, and then the darker pink means that there's more than 1. So for example, in this diffuse midline glioma, there was more than 1 TP53 alteration identified, and then there was only 1 of the additional genes. So another thing to notice from this table is that as you move from low grade to high grade, there are more oncogenic alterations that accumulate as you get higher grade tumors. So now I'm going to talk about the alteration types in more detail, starting with the single nucleotide variants. So again, there were not as many as perhaps one might expect in our data. So we had some pretty common FGFR1 single nucleotide variants, ones that have been described and have been proven to be oncogenic variants. So we had 7 tumors that had the N546K alteration in FGFR1, and then we had 1 with a 656E, K656E in a pilocytic astrocytoma. And for the tyrosine kinase duplications, there was a good number of these, and again, these were only seen in FGFR1. There was not a whole lot of variability in the breakpoints, but one important thing to note is that the breakpoints did encompass the entire tyrosine kinase domain each time, which spans exons 11 through 17. So the 5' breakpoint was anywhere from exon 9 to 11, but in any case, it spanned the entire tyrosine kinase domain, resulting in a full duplication. And this duplication has, in fact, been shown many times over to be oncogenic. And there are several papers that describe this, but this is one that I think described it nicely, just to prove that this is relevant. This does cause oncogenicity, and it leads to activation of the MAPK pathways. It's also one of the most frequently observed FGFR aberrations in pediatric low-grade gliomas, which is consistent with what we found. The next alteration type I want to discuss is fusions, and there was a lot more diversity in the fusion types, and specifically lots of different partners besides FGFR. So I only picked a couple that I felt like were perhaps the most relevant, and the first one I'll talk to you about is FGFR1 TAC1. We found 2 of these, and these were both seen in pilocytic astrocytomas. Here's an example of the histology of a pilocytic astrocytoma here. And while this is not the most common alteration that you see in a pilocytic astrocytoma, it is relevant because it can fulfill the diagnostic criteria needed for a PA, which is one of the diagnostic criteria is that there needs to be a MAPK mutation. So this just highlights that FGFR alterations have a lot of utility in these tumor types, and one of which is diagnostic, not just prognostic or therapeutic. So that's one of the reasons I wanted to highlight this one. And then the other one is FGFR2, CT, and NA3, which we found in 3 plenties. And again, this is a diagnostic criterion that it's only ever been reported in plenties, so it was not unexpected to find this. Okay, so what does all this mean? Why is it important? What can we do with it? So first of all, just to recap, FGFR structural variants are more common in CNS tumors compared to the single nucleotide variants in FGFR, and that is consistent with what we found. This is especially true for the low-grade tumors, and this highlights the need for the effective structural variant detection, which is a reason that our tyrosine kinase domain duplications is one of the most important pieces of this puzzle, and that's where our detection methods really became important here, where we were able to detect these larger structural variants. And since FGFR alterations are well-known contributors to pediatric CNS tumors, they have the potential to be targeted by tyrosine kinase inhibitors. And in fact, like I think we all heard from Dr. Parsons' wonderful presentation earlier, there are a lot of drugs out there that do target these alterations, but they're in either a different tumor type or they're only in adults. So there is a limited amount of opportunity for children to be enrolled in these trials. Of course, we just heard of an exception to that earlier, but there's still room for a lot more children to be potentially helped by identifying these alterations and then potentially having a therapeutic option for that. So lastly, I want to thank all of you for your attention, and I'd like to thank everyone who helped and contributed to this body of work. So all of the people who work in the lab and the clinicians involved and all of the other pathologists. So thanks to everybody, and with that, I'm happy to take any questions or comments, or we're waiting till the end, right?
Yep.
Okay, well, save your questions for later, but thank you, everyone.